Hello and welcome to week 9. You have made it halfway through the course. Congrats, that's me clapping for you. Honestly, in my opinion, the second half of the course is much more fun than the first half of the course. So I hope you like it, because what we're going to be talking about is all the stuff that actually explains how it is we look the way we do and how it is our cells actually operate. So we're going to start by focusing in on DNA and the molecular basis of inheritance. We're going to start with a little history and thinking about, well, how did we figure out all the stuff that we're about to learn? How was it determined that DNA is the genetic material passed down from our parents, giving us the code for making our bodies? So we're going to learn later in the class about Charles Darwin and Gregor Mendel, but now we're going to be focusing on the history of DNA. So we have T.H. Morgan's group showing us that genes are on chromosomes, but that chromosomes have different components, DNA and proteins. And it really was thought at this time, back in the early 1900s, that it was the proteins that had to have been the genetic material, because proteins, there's lots of different types, they're interesting, whereas DNA is pretty much the same. There's only 4 different bases. It's just a boring polymer. So people were really interested in what actually made up this genetic material. So one of the first amazing experiments was done by Frederick Griffith in 1928. And what he was doing is he was studying different types of pneumonia, a pneumococcus, so a bacteria. And he had one strain that was smooth, that was able to kill the mice. So this was a lethal pathogenic strain, smooth cells that had this capsid. He had another strain that was called rough that didn't have that capsid, and that strain wasn't lethal. It didn't kill mice. He also could show that if you took that lethal strain and heated it, so he broke down the bonds of the proteins and some of the lipids, that bacteria would no longer be lethal, so that those mice injected with heat-killed S cells, they lived. But then he did an interesting experiment. He combined the heat-killed bacteria with the living bacteria, and he showed that those mice injected with both died. And while he could have concluded that something he did brought back the heat-killed bacteria to life, because when he would culture those mice, he could find the smooth S strain. But what he instead concluded, and has history and biology has proved, was that instead something was being passed from this heat-killed pathogenic strain to that living strain, that there was this transforming principle. But it wasn't until Oswald Avery and Colin MacLeod and McCarthy that they actually showed that it was specifically the DNA that was that transforming principle. But even though they showed this in 1944, not everyone believed them. Erwin Chargaff continued to do research into DNA and actually had found by early 1950 that the composition of bases within a particular species differs from the composition of DNA in a different species. Meaning if you add up how much adenine, guanine, cytosine, and thymosine are in yeast or a rat or a human, they're going to be different amounts. But very intriguing and interesting, he also showed the amount of adenine equals the amount of thymine, and the amount of guanine equals the amount of cytosine. Now you would think that this would be instantly make sense that adenine must pair with thymine and guanine must pair with cytosine. But that's not at all how this data was interpreted. In fact, it wasn't until 1952 that really the scientific community accepted that it was actually the DNA rather than the protein that was this transforming principle. So what did Alfred Hershey and Martha Chase show? So you can watch this video or you can listen to me. What they did is they knew that there was bacteriophage. They knew that there was viruses that could infect bacteria and injected something into that cell. And they knew that that something must be that transforming principle. So what they did is they labeled either the radioactive sulfur, which of course you remember sulfur is in the amino acids of cytosine and also methionine. And so sulfur will label just proteins. There is no sulfur in DNA. So they took radioactive sulfur and they labeled their viruses. And then they took those radioactive viruses and then they saw, could they isolate that radioactive protein in the infected bacteria cells? So what they did again is they labeled their phages, so that the radioactive sulfur, they infected the cells. Then they shook up the cells in the bacteria to separate the bacteria from all the stuff that was outside. And then they determined where was the radioactivity. Was it inside the cell in the pellet or outside in the liquid? And when they labeled sulfur and proteins, they found that radioactivity remained outside the cell. But when they instead used radioactive phosphorus, that of course is going to label all the DNA. And then they injected, same thing, they labeled the proteins, and then they followed the same experiment where they took that labeled phage and they infected cells. They then broke up the cells from that viral particles that were remained outside, so that weren't injected in, and then they were able to isolate and identify that inside that bacteria that was infected, it was the DNA that had been passed down, definitively showing that it is DNA that is the transforming principle and the basis for our inheritance. So once it was accepted that DNA was the transforming principle, the structure of DNA becomes super important. Now you're all going to be watching a video on the history of the structure of DNA. You get to learn about the role of Rosalind Franklin, which made this picture, which really actually shows the structure of DNA, and nobody could have solved the exact structure of DNA without her careful work that was reviewed without her knowledge, and led to a Nobel Prize by Maurice Wilkins and James Watson and Francis Crick. So we are actually familiar with the structure of DNA because we've already been learning about it, but again, we will review. There are RNA and DNA nucleotides. They each have 3 parts. They have a sugar, which is different. It's either deoxyribo or ribose. They've got bases, a nitrogenous base, meaning it's full of nitrogens, and phosphate groups. So you've got the 3 components of a nucleotide. Now, there's different nitrogenous bases. For DNA, there's guanine, cytosine, thymine, and adenine. In RNA, we're going to see that there is not a thymine, there is instead uracil. Now, I'm also going to comment right here, we can see the structure of these sugars. You can see that there's an oxygen. As you'll learn in organic chemistry, that you can order the carbons in a particular order, but it's always going to start right to the right of that oxygen. So here's the number 1 carbon, 2, 3, 4, 5. Remember, ribose is a 5-carbon sugar as opposed to glucose, which is a 6-carbon sugar. We can also talk about the differences in bases, and we can put them into different groups. There are pyrimidines. These are these single ring nitrogenous bases. So that's cytosine, thymine, and uracil. And here again, you can see that difference between thymine and uracil. Really, it's just this methyl group. You can also see the purines. The purines have these double ring structures. So that's adenine and guanine. And for what it's worth, I always remember that purines is a shorter word, and so it's the longer, it's the bigger structures, where pyrimidines is this longer word, which has 3 different components, but they're only single structures. So any little mnemonic tricks you can do like that will be helpful. But again, we have the components of a nucleotide. We got that nitrogenous base, we have that sugar, and then we have a phosphate group. And what we see in DNA and RNA is that there's the sugar phosphate backbone with the nitrogenous bases on the side. And that makes the famous double helix that we see in DNA, where you have 2 different strands running in opposite directions. Again, we can pay attention to that numbering of the carbons, where you're going to have a 5' end, meaning the 5 carbon, which is right here, which is attached to the phosphate group, can be ordered on this side. It means the other end will always be the 3' end. And why it's called the 3' end, because the 1, 2, 3, The third carbon is right here on the very end. Now, the double helix is going to run in opposite directions. If the 5' end is on the top, the opposite strand is going to have its 3' end. Again, its third carbon here with its 5th carbon right here. So they run in opposite directions and they have direction. There's directionality to these. And in DNA, those nitrogenous bases form hydrogen bonds with each other, with adenine binding with thymine and guanine binding with cytosine. So then you want to ask yourself, can you identify a monomer? Do you understand which phosphate group is attached to which sugar? So hopefully you can identify either like this. So a phosphate group is always going to go with the 5' carbon. You can also use this graphic. But you need to understand this numbering of the carbons and the structure of DNA with the 5' carbon bond into a phosphate group, which then it's going to bind the next sugar at the 3' carbon. So this phosphate group will find a bond with this 3' carbon in the deoxyribose. So you have a 3' end and a 5' end. So you can watch these videos. You need to be able to know everything about this structure of DNA. So I've got several more pictures showing you the same key information, that you've got hydrogen bonds, and those are going to be important, that are much easier to break than the covalent bonds formed between the 3' carbon of a sugar and that phosphate group. So you have 5' ends and 3' ends. And we're going to see in the next PowerPoint the importance of having these free 3' ends available to be adding nucleotides. So we talked all about that double helix of DNA with its anti-parallel, meaning going 2 different directions. And I also want to point out RNA. RNA is single-stranded. It will still fold up on itself and form structures and form nitrogenous base pairs, but it is in fact one strand that contains uracil and ribose. And now you can give yourself a little quiz. Again, can you identify one nucleotide? Do you know if this is RNA or DNA? And then what would you change? So of course, one nucleotide, it's going to have the phosphate group from the 5' end. So the phosphate goes with the 5' end sugar. It's going to bond with that 3' carbon of the next sugar. In this case, we know that it's DNA because the sugar is deoxyribose and there is a thymine. If I wanted to make RNA, I would need to have uracil and I would need to have it to be ribose and adding an oxygen. So that's the structure of DNA and RNA. But now we can think about how are they structured in the cell? And that is into what we call chromosomes. Now, typically you picture them like this, as this sort of little bit of a X shape, but this is actually only after they've been replicated. So how these chromosomes form is that you take that DNA, and that DNA gets wrapped around proteins called histones. Now, DNA is negative based by all those phosphate groups, and those proteins are positively charged. So they wrap tightly around, and then it's kind of like a phone cord that gets super duper tangled. So we have 6 feet of DNA in each one of our cells. So in something smaller than the point at the end of a sentence, we have 6 feet of DNA. So how do we get that much DNA in such a tiny structure? It's that it's highly, highly condensed. So the DNA gets wrapped around these proteins. So it gets 2 wraps for each histone. Then those histones all wrap together and forming this condensed form of DNA, which is chromatin. So chromatin is this mixture of DNA and proteins all wrapped up together tightly. Then this fiber gets wrapped, and then that fiber gets wrapped, ultimately making that chromosome. I highly recommend you guys check out this site. I'm going to see if it's going to work right now, but I highly recommend you check out this site. And what you can do here is you can play around with DNA and the structure and seeing how these different parts come together. You can see those 6 different carbons. You can see how DNA actually replicates. So I highly recommend checking out this page. but unfortunately you do need to use the flash in order to use it. So back to our picture, we have chromosomes, which are highly condensed chromatin, which are that DNA wrapped in all this hard structure. We can also dive into our human genome. So our human genome is 46 pieces of DNA, which are actually 23 pairs of chromosomes. And we're going to spend a lot more time on this in the coming chapters, but I wanted to sort of introduce you and give you a little bit of the wow here. So here again, we're seeing these replicated chromosomes where they've got 2 copies. And again, that's all next section. So most of the time, actually, though, chromosomes are just that single strand. And we can think about how much DNA is in us. So if this plays right now, it would take 5 years watching this speed of nucleotides to complete the entire human genome. I'm not going to read all of these pairs, but the next few slides are just telling you our human genome has got so many nucleotides. Apparently a 3-year daily subscription to the New York Times. Or again, I'm not going to read all of these, but just to give you some idea of how many nucleotides are in our genome. And then what's shocking is how much of our DNA is actually a gene and is something that we think is important. So that is the end of part 1 of this week.